Biochemistry. Enzymology is a branch of biochemistry that studies enzymes, which are proteins that function as catalysts in biological chemical reactions. Enzymes accelerate the rate of chemical reactions in the body without being changed in the reaction. This process is very important because many chemical reactions in the body occur very slowly without the help of enzymes. Here is a detailed explanation of enzymology. Characteristics of enzymes. Enzymes have several characteristics that distinguish them from ordinary chemical catalysts. A specificity. Namely, enzymes have high specificity towards substrates, molecules converted by enzymes. For example, amylase will only convert starch into sugar, while lipase only converts lipids. B. Reaction speed, namely, enzymes can accelerate chemical reactions up to millions of times faster than without enzymes. C. Adjustable activity, namely, enzyme activity can be influenced by factors such as pH, temperature, substrate concentration, and the presence of inhibitors or activators. D. Regeneration, namely, after playing a role in the reaction, the enzyme is not degraded or permanently changed, so it can be reused for the next reaction. 2. Enzyme structure. Enzymes are proteins consisting of one or more polypeptide chains. The structure of the enzyme is divided into two main parts. A. Active site. This is the site on the enzyme that binds to the substrate and where the chemical reaction occurs. This part has a very specific three-dimensional shape and can only bind to certain substrates, according to the lock and key theory. B. Allosteric site, namely, some enzymes have allosteric sites, which are binding sites for regulatory molecules, inhibitors or activators. Binding of molecules to allosteric sites can change the shape of the enzyme and affect its activity. 3. Enzyme working mechanism. Enzymes accelerate chemical reactions by lowering the activation energy, the energy required to start a reaction. This is achieved in several ways. A. Decreasing activation energy, namely, enzymes reduce the distance between the substrate and the product, so that the reaction can take place faster. B. Transition stability, namely, enzymes can stabilize the transition state of the reacting substrate, so that the process takes place more efficiently. C. Substrate orientation, namely, enzymes facilitate the orientation of the substrate in a more appropriate position to react. D. Catalytic function, namely, some enzymes use functional groups in their active sites, e.g., amino, hydroxyl or sulfhydryl groups, to play a direct role in chemical reactions. IV. Enzyme kinetics. Enzyme kinetics is the study of the rates of reactions catalyzed by enzymes and the factors that affect those rates. A model often used to describe enzyme kinetics is the Michaelis-Menten model. A. Michaelis-Menten model. This model describes the relationship between substrate concentration and enzyme reaction rate. In this model, there are two main steps. 1. Formation of enzyme substrate, S complex, namely, the substrate binds to the enzyme to form a temporary complex. 2. Catalysis, namely, the S complex leads to the formation of products and the release of enzymes. The rate of enzyme reaction can be described by the Michaelis-Menten equation. V equals frac V underscore text max S K underscore M plus S, where V is the reaction rate. V underscore max is the maximum reaction rate when all active sites of the enzyme are filled by the substrate. S is the substrate concentration. K underscore M is the Michaelis constant, which describes the affinity of the enzyme for the substrate, the smaller the K underscore M. The higher the affinity of the enzyme for the substrate. B. Factors affecting enzyme reaction rate. Some factors that affect the rate of enzyme reaction are 1. Substrate concentration, namely, the higher the substrate concentration, the faster the reaction rate until it reaches V underscore max. 2. Enzyme concentration, namely, the more enzymes, the faster the reaction rate. Because more active sites are available. 3. pH, namely, each enzyme has an optimal pH where the enzyme works most efficiently. A pH that is too high or too low can damage the enzyme structure. 4. Temperature, namely, high temperatures can increase the reaction rate to a certain point, but temperatures that are too high can damage the enzyme structure, denaturation. 5. Inhibitors, namely, molecules that can reduce enzyme activity. Inhibitors can be competitive, compete with the substrate to bind the active site, or non-competitive, bind to sites other than the active site and change the shape of the enzyme. 6. Activators, namely, molecules that increase enzyme activity by binding to the enzyme or increasing its affinity for the substrate. Versus types of enzymes. Enzymes can be classified into several classes based on the type of reaction they catalyze. 1. Oxidoreductase, namely, catalyzes redox reactions, electron transfer. 2. Transferase, namely, catalyzes the transfer of chemical groups, e.g., methyl, acetyl, from one molecule to another. 3. Hydrolase, namely, catalyzes the breaking of chemical bonds using water. 4. Lyases, namely, catalyzing the breaking of bonds without using water or atom transfer. 5. Isomerases, namely, catalyzing changes in molecular structure without adding or removing atoms. 6. Ligases, namely, catalyzing the formation of new bonds, usually accompanied by the use of energy from ATP. V. Regulation of enzyme activity. Enzyme activity can be regulated by several mechanisms. A. Allosteric regulation, namely, regulatory molecules bind to allosteric sites, which changes the conformation of the enzyme and affects its activity. B. Cofactor modification, namely, some enzymes require cofactors or coenzymes, e.g., metal ions or vitamins, to function properly. C. Genetic regulation, namely, the expression of genes that code for enzymes can be regulated, so that enzyme production increases or decreases according to cell needs. D. Inhibition and activation by other molecules, namely, other molecules can activate or inhibit enzyme activity to control certain metabolic pathways. Conclusion. Enzymology is an important study in biochemistry that provides deep insight into how enzymes work as catalysts in biological reactions. Enzymes speed up chemical reactions, regulate various biological processes, and have various regulatory mechanisms to ensure that cell metabolism runs efficiently and in a controlled manner.